Credit to Chapter 4 Fungi. Fungi are achlorophyllous, heterotrophic, eukaryotic, and spore-bearing organisms whose cell wall is made up of an chitin or cellulose or both, which reproduce by the asexual and the sexual spores method. The fungi are too diverse and may be easily defined. Fungi obtain food by the absorption except from the lower group which obtain food by ingestion. There are something 5,100 genera and more than 250,000 species of fungi. Study of fungi is known as mycology. They are avascular, typically non-motile. They absorb or facultative anaerobes, organoheterotrophic mode of nutrition, and may be saprophyte, which is which obtain the food from the dead material, or some are parasite which remain attached to the other organism for the obtaining the food. General characteristics of fungi are they all are eukaryotic, possess membrane-bound nuclei, and the large range of membrane-bound cytoplasmic organelles, for example, mitochondria, vacuoles, endoplasmic reticulum, etc. Most of them are filamentous. Compound of an individual microscopic filament is called as an hyphae. So individual filament is a hyphae which exhibit apical growth and which branch to form a network like hyphae called as a mycelium. Some are unicellular, for example is in yeast. Protoplasm of an hyphae or cell is surrounded by a rigid wall composed primarily of an chitin and glucans, although the walls of some species contain cellulose. Many reproduce both sexually and asexually. Both sexual and asexual reproduction often result in the production of spores. Their nuclei are typically haploid and hypal compartment are often multinucleated, although the oomycota and some yeast possess diploid nuclei. Fungi are achlorophyllous. They lack chlorophyll pigment and are incapable for the photosynthesis. The mode of nutrition hence is a heterotrophic mode of nutrition. They utilize a pre-existing organic source of carbon in their environment and energy from the chemical reaction to synthesize the organic compound they require for the growth and energy. They possess a characteristic range of storage compound, for example, trehalose, glycogen, sugar, alcohol, and lipids. May be free living or may form an intimate relationship with the other organism, that is, may be free living, parasitic, or mutualistic, like a symbiotic association in a lichen. So the plant body of a fungus is called as a thallus, which is a unicellular or the multicellular. There are the two types of thallus, eucarpic and holocarpic. Eucarpic thallus is differentiated into the vegetative part which absorbs the nutrient and the reproductive part which forms reproductive structure. Such thalla is called as an eucarpic. For example, is an pythium. Holocarpic thallus does not show any differentiation of vegetative and reproductive structure. After a phase of vegetative growth, it gets converted into one or more reproductive structure. Such thalli are known as holocarpic. For example, is a Saccharomyces yeast, etc. Hyphae is a tubular, transparent, and filament, usually branched, composed of an outer wall and the cavity known as a lumen, lineate, or filled with the protoplasm, including cytoplasm. Hyphae are divided into the compartment or the cell by the cross wall called septa and are generally called a septate hyphae or sinusoidal or septate without cross wall. Hyphae of the most of the fungi measure from the 5 to 10 micrometer in length. The mycelium, the hypal mass or the network of an hyphae constituting the body of the fungus is called as a mycelium. So mycelium is used to obtain the food from the other plant or the other host plant body. They obtain the nutrient through the host area. There are the two types of an mycelium, monokaryotic mycelium and dikaryotic mycelium. So monokaryotic mycelium is a uninucleated mycelium containing single nucleus that usually form the part of an haplophase in the life cycle of an fungi. Dikaryotic mycelium is binucleated mycelium contains pair of a nuclei, that is dikaryon, which denotes the diplophase of the life cycle of a fungi. So there are the different types of fungi: septate and cynocytic fungi, and aspergillus is an example of an fungi. They are the dimorphic fungi. Most serious fungal pathogens grow as an yeast or the molds, depend upon the environmental condition. The fungi are typically reproduced by the means of spores. Sexual meiotic and asexual mitotic spores may be produced. Asexual fragmentation may occur. Spores are typically released from the aerial hyphae in molds. Vegetative hyphae are involved in the nutrient absorption. Asexual spores typically bind, disperse, and commonly used in the identified fungal pathogens. Two types of asexual spores are present: sporangia spore inside the sporangium sac, and conidiospore, that is a various form not formed in a sac. So these are the conidiospores. Sporangiospores present in the rhizopus. Most of the yeast reproduce asexually through the body. From the pseudohyphae or Saccharomyces is the example. Fusion yeast divides evenly to produce two cells. Saccharomyces is the example. So classification of an fungi. The kingdom fungi is divided into the two divisions, namely Myxomycota and Eumycota. Myxomycota means presence of an plasmodium or the pseudoplasmodium, and Eumycota means absence of an plasmodium or pseudoplasmodium. So fungi are divided into Myxomycota and Eumycota. Division: Myxomycota includes four classes as Acrasiomycetes, Labyrintholates, Myxomycetes, and Plasmodiophoromycetes. So Acrasiomycetes, these are the free living excretory phase of an amniotic unit or as an pseudoplasmodium before the production. Labyrintholates are the net plasmodium is present. Myxomycetes, presence of a free living saprobic plasmodium. Plasmodiophoromycetes, presence of an parasitic plasmodium within the host cell. Second division is a Eumycota. There are the 5 subdivisions including subclasses. Subdivision Mastigomycotina, in which zoospores present. Perfect state spores are typically oospores. Zygomycotina, zygospores absent. Perfect state spores are zygospores. Ascomycotina, zoospores and zygospores absent. Perfect state of spores is ascospores. Basidiomycotina, zoospore and zygospores absent. Perfect state of spores are basidiospores. Deuteromycotina, zoospores and the perfect state spores like in zygospores, ascospores and basidiospores are absent. Subdivision Mastigomycotina, it includes 3 classes: Candidiomacetes, Hypochondriomycetes, and Oomycetes. Candidiomacetes are the zoospores with the whiplash types of flagella. Hypochondriomycetes, zoospores with the tinsel type of flagellum, and Oomycetes, zoospores with the biflagellate. Subdivision Zygomycotina, it includes 2 classes: Zygomycetes and Trichomycetes. So zygomycetes are mostly saprobic, and trichomycetes are attached to the cuticle or the digestive tract of an arthropods. Subdivision Ascomycotina, it includes 6 classes. First one is the Hemiascomycetes, in which Ascogenus hyphae and Ascoscarp is absent. Loculoascomycetes, in which Ascogenus hyphae and Ascoscarp is present. Platomycetes, Ascoscarp accepted. Labilbineomycetes ascoscarp of an perithecium type. Pyrenomycetes ascoscarp typically perithecium. Discomycetes ascoscarp apothecium. Subdivision Basidiomycotina. It includes three classes: Teleomycetes, in which basidiospores absent, parasitic on vascular plant; Hymenomycetes, mostly saprophytic, basidiocarp is present; Gastromycetes, mostly saprophytic, basidiocarp angiocarpus. Subdivision Deuteromycotina. It includes three classes: Blastomycetes, that is, in true mycelium is poorly developed or absent; Hypomycetes, true mycelium well developed; Coelomycetes, budding cell are absent, spores develop in a cervili in or pachynidia.